enzymes, that is also known as biocatalyst. And without enzymes, no biochemical reactions can occur inside our body. And this enzyme, which is also known as the biocatalysts, can be classified into 6 different categories. And in this particular video, I am going to discuss about these 6 categories of the enzyme based on the function they perform. In our body, enzymes are considered generally one of the most important biomolecules because without enzymes, each and every second, the different biochemical reactions that are continuously being happening inside our body will not execute it. So therefore, enzymes are very, very much important in our body in order to survive. Now, the most fastest enzyme in our body is the carbonic anhydrase. Carbonic anhydrase is the most fastest enzyme. What does it do? Carbonic anhydrase catalyzes the production of carbonic acid, that is H2CO3. And this production of carbonic acid H2CO3 from carbon dioxide and water is the fastest reaction that any organism has in their body. In a single second, around 10 to the power 6 molecules of carbon dioxide reacts with water to produce carbonic acid. So now come back to the different types of enzyme that will be discussed in this video based on the function they perform. So enzymes can be classified into 6 basic categories. The first one is the oxidoreductase. As the name implies, oxidoreductase means the group of enzyme that will perform the oxidation and reduction of their substrate. The enzymes that participate in the oxidation or reduction of their substrate. Each and every enzyme has a specific type of substrate upon which it acts on. So this particular group of enzymes will either remove electron or add electron. We all know that removal of electron means oxidation and addition of electron means reduction. Or in terms of hydrogen, removal of hydrogen, that means oxidation, and addition of hydrogen, that means reduction. So this hydrogen, electron, or in terms of oxygen, it can be added or it can be removed from their substrate. So one substrate molecule will be reduced, another substrate molecule will be oxidized by this particular group of enzymes. So enzymes which catalyze oxidation and reduction between the two substrates are classified as oxidoreductase group. So here the substrate which is in the reduced configuration and another substrate is prime which is in the oxidized configuration, and these two substrates will interchange their oxidation state by the help of this oxidoreductase group of enzyme. And this oxidoreductase group of enzyme is also known as dehydrogenase. Dehydrogenase, whenever you will find any enzyme ends with dehydrogenase, definitely the enzyme is involved in the different kind of oxidation reduction reaction. So the S substrate which was in the reduced state is now in the oxidized condition, and the S prime substrate which was in the oxidized state is in the reduced condition. So oxidoreductase group of enzymes oxidizes or reduces their substrate. Some of the examples of oxidoreductase enzymes definitely will end by dehydrogenase. For example, lactate dehydrogenase, pyruvate dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase. So any enzyme that ends with dehydrogenase is definitely an oxidoreductase group of enzyme. Now next class of enzyme is a transferase. Next group of enzyme is a transferase. Transferase group of enzyme transfer a particular type of group from one substrate to another substrate. It can be phosphate, it can be hydroxyl group, or it can be any other functional group. So it will only transfer a particular functional group from one substrate to the next substrate. For example, here the substrate with a functional group G, another substrate which is not with any kind of functional group. So this G will be transferred to the S prime, and the S prime is now linked with G, and the S is devoid of any kind of functional group. So the transferase are the group of enzymes that catalyzes a transfer of group. One group, it can be alcoholic group, hydroxyl group, aldehyde group, it can be phosphate group, whatever the group can be, it will be transferred from one substrate to the next. Next group of enzyme is the hydrolase. As the name hydrolase, so definitely it will hydrolyze their substrate. It will help in the broken up of their substrate into multiple products with water, in presence of water. Yes, hydrolases will never act in absence of water. Without water, hydrolase can never act upon their substrate. So in presence of water, the enzymes which are under hydrolase category will cleave the different types of bond. What are the different types of bond? It can be ester bond, it can be ether bond, it can be peptide bond, or it can be glycosidic bond. These are the different types of bonds that can be cleaved by the enzyme under hydrolase category. But all of this cleaving will definitely need water. Without water, this cleaving will not be achieved. So what are the other different types of bond that can be broken up? Carbon-carbon bond, carbon halide bond, phosphorus nitrogen bond, and these all of the type of bond you have to memorize. In the MCQ exam, in the NEET exam, these bonds will be mentioned. What are the different types of bond that can be cleaved by hydrolase group of enzyme? The first one ester, then the ether, then the third one peptide bond, then the glycosidic bond, carbon-carbon bond, carbon halide bond, or the PN bond. So they will hydrolyze their substrate. Next, already we have discussed the three group of enzyme. The first one oxidoreductase, second one transferase, third one hydrolase. Now comes to the fourth one, that is lyase. Fourth one, that is the lyase. The function of lyase is similar like the hydrolase, but the lyase will act over their substrate without water. In absence of water, it does not need the presence of water in order to execute its function. So lyase do the same or similar function like the hydrolase, but quote unquote without water. Kono rokom water lage na. So enzymes that catalyze removal of groups from substrates by mechanisms other than hydrolysis. Kono rokom hydrolysis hobe na. Tar mane without water and leaving double bond. So enzyme কোন ধরনের enzyme double bond form করে? কোন ধরনের group of enzymes double bond form করে? That is the lyase, যে double bond form করবে। আর removal of group কে করে? Removal of group lyase. Removal of group, মানে লাইজের যদি আমরা ক্যারেক্টার দেখি, টোটাল তিনটে মেজর ক্যারেক্টার আমরা লাইজের মধ্যে দেখবো। এক নম্বর আমরা দেখলাম রিমুভাল অফ গ্রুপ, সেকেন্ড ওয়ান আমরা দেখলাম আদার দেন হাইড্রোলিসিস, ভাঙবে রিমুভাল অফ গ্রুপ আদার দেন হাইড্রোলিসিস। এই আদার দেন হাইড্রোলিসিস কথাটার মানেই হলো উইদাউট ওয়াটার। আর একটা দেখলাম ডাবল বন্ড ফর্ম করবে। দেখো এখানে সিসি এর মধ্যে একটা এক্স একটা হচ্ছে ওয়াই, দুটো সাব ইউনিট আছে। তো এখানে এক্স এন্ড ওয়াইটা সি থেকে আলাদা হয়ে গেল, এন্ড সিসি এর মধ্যে সিঙ্গেল বন্ড ছিল, এখানে এখন সিসি এর মধ্যে কিন্তু ডাবল বন্ড তৈরি হলো। ডাবল বন্ড কোন এনজাইম তৈরি করে? ডাবল বন্ড তৈরি করে কিন্তু লাইস। নেক্সট কামস টু দ্য নেক্সট গ্রুপ অফ এনজাইম, দ্য ফিফথ গ্রুপ অফ এনজাইম, that is isomerase. The fifth group of enzyme, that is isomerase. Isomerase, as the name implies, isomerase will form isomer of their substrate. What are isomers? Isomers are structurally not similar, but functionally they are identical. Isomers have some different functional group in their molecular structure, but they have the same molecular formula. For example, glucose and fructose. Both of the monosaccharide has same molecular formula C6H12O6. But if you look upon the internal molecular framework of glucose and fructose, on glucose the first carbon has an aldehyde group, but in case of fructose the second carbon has a keto group. So glucose is considered as aldo sugar or aldohexose, but fructose is considered as ketohexose. But both of them have the similar formula C6H12O6. So glucose and fructose are aldo-keto isomer of each other. So this isomer production, this isomer formation will be done by this group of enzymes, that is isomerase. So they include all the enzymes catalyzing the interconversion of optically geometric positional of the isomer. Optically এই লাইনটা তোমাদেরকে মনে রাখতে হবে, optical geometric position of the isomer. এটা হচ্ছে তোমরা যখন হায়ার স্টাডিজ করবে তখন তোমরা দেখতে পাবে দু রকমের অপটিক্যালি কার্বোহাইড্রেটস হয় বা দু রকম অপটিক্যালি অ্যাক্টিভ monosaccharides হয়. One of them is the D isomer, আর একটা হয় হচ্ছে L isomer. D আর একটা হলো হচ্ছে L. D stands for dextrorotatory and L stands for levorotatory. D stands for wherever the plane polarized light is passing through the fluid solution, it will bend the light towards the clockwise. It will bend the light clockwise direction. তাহলে সেটা হবে dextrorotatory, and if the direction of the plane polarized light is bent in anti-clockwise direction, then the sugar will be termed as levorotatory. So this dextrorotatory or levorotatory optical isomer is produced by this group of isomers, but they have the same molecular formula. Next, ligases. The sixth one is the ligases. Ligases means they will ligate, they will join one substrate, another substrate. They will join up both of the substrate forming a single one. দুটো সাবস্ট্রেটকে জয়েন করবে এনজাইমস ক্যাটালাইজিং দ্য লিঙ্কিং টুগেদার অফ টু কম্পাউন্ড. দুটো কম্পাউন্ডকে একসঙ্গে জয়েন করবে, দুটো কম্পাউন্ডকে একসঙ্গে জয়েন করে একটা কম্পাউন্ড তৈরি করবে. যেমন জয়নিং দ্য CO বন্ড, CS বন্ড, CN বন্ড, PO বন্ড. এই সমস্ত বন্ডগুলো কার্বনের সঙ্গে অক্সিজেন, কার্বনের সঙ্গে সালফার, কার্বনের সঙ্গে নাইট্রোজেন অ্যান্ড কার্বনের সঙ্গে অক্সিজেন, এই বন্ডগুলো ফর্ম করে কিন্তু দুটো কম্পাউন্ডকে জয়েন করবে. সো দিস আর দ্য 6 মেজর গ্রুপ অফ enzymes based on the different types of reaction they catalyze. Oxidoreductase, next আমরা দেখলাম transferase, 3 নম্বর আমরা দেখলাম hydrolase, 4 নম্বর আমরা দেখলাম lyase, 5 নম্বর আমরা দেখলাম isomerase, and the 6th one, that is the ligase. So definitely this video will be very much helpful for understanding the classification of enzymes, and if you still have any doubt, please let me know in the comment box. I will definitely get back to you. Tata.